So for the first lecture in this week, we'll be on the cell cycle. And often it'll be called mitosis, which is technically not correct. So what we're trying to do is look at divisions. And here we're seeing 2 cells turn into 4 cells. But what we want to know, what we want you to know is there is really kind of 2 different cycles that go on in the body. One is mitosis, and we'll talk about that in the short session. And then later we will talk about meiosis. Mitosis, the idea behind this is to take 1 cell and to get 2 identical daughter cells. And then meiosis starts with 1 cell. And then what you do is you end up with 4 cells that are genetically different than each other. And so they both have different purposes. And so we will start with mitosis. And to do that, really, though, we call it the cell cycle. And so this is really showing you the parent cell going into the 2 identical daughter cells. But notice that on the left here, we don't call it just mitosis. It's really interphase, mitosis, and cytokinesis. There's 3 different steps. And so often when mitosis has been taught, may still be taught, I'm not sure what people are doing out there, but reality, often you would hear IPMAT as kind of your easy, cheap way to memorize the stages of mitosis. But interphase technically is not part of mitosis. And there's also now proto-metaphase. So we've changed things up a bit. We've got more details on what we're looking at, and so we want to go through that stuff. But we want to start with some basics. So the key roles of mitosis is asexual reproduction, growth and development, and tissue renewal. So what we're trying to do is just make more cells, and that's pretty simple. And so then we need to talk about cellular organization of the genetic material. And you're going to hear terms like chromatin, chromatids, chromosomes, centromeres, kinetochores. So lots of terminology, and you want to make sure that you have all of that pretty straight in your head as you go into a quiz or a test, because it can be a tad bit confusing if not. So when it's all spread out, it is in the form of chromatin. And then when it tightens up and condenses, they're called chromosomes, although technically they are still made of chromatin. So that becomes a little difficult. Okay, so here we go. This is when they're condensed, this picture here. And so what I'm looking at is one that has gone through DNA replication, what we talked about last week. And so this one is a chromosome made up of 2 sister chromatids. Those are identical information. And those sister chromatids are held together by what we call centromeres. And each one of those centromeres actually has is tied to one of the chromatids. I'm telling you this because the terminology is changing a little bit from before. And it's a little worrisome to me that you might get it confused, because what they would always say is just 2 sister chromatids held together by a centromere. What they're finding out now is that repetitive DNA structure is going to be called a centromere, and they're actually considered them a little separate pieces. So technically, they have a centromere that's held together, and often in explanations, you'll hear it as a single centromere. So that can be a little bit confusing. All right, so we want to talk about the cell cycle before we move on. So again, instead of it all being just mitosis, what we're looking at here is we have interphase, inter means between, so it's in between meiotic divisions. And then you have mitosis, which is talking about what's happening in the nucleus of a eukaryotic cell. That is the process with the steps that we'll go over. And then cytokinesis is actually talking about what's happening to the cell at the very end of that time. It's usually associated with the mitotic phase, telophase. And so kind of three different parts, I guess. And then interphase is actually divided into what you see is the G phase, the G1 phase, S phase, and G2 phase. And we'll talk about that a little bit more in detail. And so that's kind of the basics. And what we'll do now is we'll go through mitosis and the different steps.